All right, we'll get started. So welcome everyone. I'm Allie Keene with the New York Stem Cell Foundation Research Institute. Welcome to our virtual classroom. We are excited to have you here with us today. A quick reminder that we have a number of other upcoming virtual events with more being added each week. So please be sure to check back to nicef.org often. I'll also share a link later on so you don't have to remember how to spell NICEF, don't worry. The New York Stem Cell Foundation Research Institute, or NICEF as we like to call it, is located in New York City. At NICEF, we study human stem cells in many ways to help us better understand and treat different diseases like Alzheimer's, Parkinson's, cancer, diabetes, and many more. Dr. Rekha Iyer, NICEF's Associate Vice President of Scientific Outreach, is going to teach you all about stem cells and their capabilities shortly. We welcome you to ask questions all throughout Dr. Iyer's presentation. You can do this in two ways, by submitting your questions in the Q&A chat or using the raise your hand tool that is located at the bottom of your screen. This will let us know you would like to ask a question out loud. I'll let you know when your mic is unmuted and you can proceed to ask your question. You will see a few of us answering questions in the Q&A chat and marking which ones Dr. Iyer is going to answer live. So if you see that, that's what that is. Now I'm excited to introduce Dr. Rekha Iyer.
Thanks, Allie, and hi everybody. It's really great to be here and to have all of you here, so to speak. I've been at NICEF now for about 2 years. I'm a geneticist by training, and here at NICEF I help communicate to people like you about all of the exciting science that we're doing. So today I'm going to tell you all about what stem cells are and why we think that they're pretty much the greatest thing for science since the internet. So what we do here at NICEF is use the power of stem cell research, which I'm going to tell you about today, to figure out how to better treat and cure the major diseases of our time. We do this through several major activities. One is through our research institute right here in Manhattan. We have an amazing team of scientists and engineers that work together every day on stem cell science, on developing technologies, and sort of putting that all together. We also have an extensive we have an extensive community of leading stem cell scientists all over the world that we give out funding to as a foundation, but we also work together with on our research that our scientists do here in the lab. That aspect of collaboration is something that we really value and is super important for making progress in science. And finally, we also have a lot of education and outreach, just like events like this, to inspire young, bright students like yourselves to get into stem cell science. We need all of the best minds we can working on this. This includes a summer internship program that we do every year that we hope you'll all apply for once you're in university. We also do a lot to support women in science and ensure that they have the tools that they need to succeed, and lots of other activities like hosting these kinds of webinars and tours when the circumstances permit. So we hope you'll be able to join us one day for one of those. So the motivation behind what we do here at NICEF is that a lot of the diseases that we know about in our society can't be effectively treated, even though we've known about them for many years. So we hear, of course, today a lot about COVID-19, the coronavirus, but that is a new disease, relatively speaking. And even with the old diseases that we've known about for 100 years, we could still be doing a whole lot better. So for example, many of you have probably heard of diabetes. We've still been treating diabetes for about 98 years since insulin was discovered, using therapy for that. We also all probably know somebody who's had cancer, and it's very challenging to treat cancer in a lot of cases too, because the standard treatments that we use aren't effective on a lot of different cancer patients. And we need to understand better why that is so we can predict the treatments that they're going to need to save their lives as quickly as possible, of course. And finally, you've probably also heard of neurological diseases or neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, multiple sclerosis, and more. And although we've also known about these diseases for a very long time, we still don't really have any therapies available that modify the disease. So that is therapies that actually stop the course of the disease, or even reverse the disease, which is what we want to achieve. And that's even after decades of knowing what these diseases are. So why is this exactly? The reason is part of the reason is that what we know in traditional medicine and what we use to treat these diseases is largely based on model organisms. So we do need to understand these diseases, and it's, we don't want to start cutting people open. That's not really the best way to do it. So we need model organisms that we can study in the lab. And what's traditionally been used in the lab to study these different diseases that I've mentioned are models like yeast, the yeast that makes your bread and wine or beer once you're old enough to drink that. Fruit flies, zebrafish, worms, and rodents. These are some of the most common model organisms that are used in the lab. And they are actually very similar to humans in many ways. We share a lot of DNA with these model organisms. Our development as, from, single cell to a full multicellular organism happens in many similar ways. And these are, of course, much easier to study in the lab than humans. And many, many different breakthroughs in our understanding of how biology works comes from these models, and lots of Nobel Prizes have been awarded for work with that. And so they've been really, really valuable tools for research. When it comes to disease, they do mimic diseases, a lot of these diseases that I mentioned and more, but it's not quite perfect. And that's because there are, unfortunately, some key differences between these different organisms and humans in terms of our biology and in terms of what causes some of these diseases. So, we use these models typically to try to discover medicines that might work for patients with these diseases. But unfortunately, what we're finding is that the medicines that we discover in these models do not always work in people. And that's because there's either, some safety issues or efficacy issues. And so when it comes to developing treatments for disease, it's really a challenging process. The process of developing a drug from when you discover a potential drug to when you're actually able to give it to patients, when your doctor is able to prescribe it to you. This is a very long process. It's very expensive, and unfortunately, it usually fails. And that includes, 80 to 90% of the drugs that we identify in a research setting, once they advance to clinical trials, which is a necessary step to test if they're safe and effective in human beings, as much as they are in these model organisms. We see that 80 to 90% of these drugs actually fail those trials. And that often happens because of safety issues, because they're not, they might be toxic for certain people or for all people, or because they're not producing the effect. They're not, treating the disease in the way that we hope that they would. And for those drugs that do make it through, it takes on average up to 15 years and billions of dollars for every drug that does actually make it through approval by the FDA, as you might have heard of the Food and Drug Administration, that has to approve all of the drugs that are available for patients here in the United States. So clearly a new approach is needed. And what the fundamental issue is that we can't treat what we don't understand. We still don't really know exactly what causes a lot of these diseases. Now, we might know how they show up in patients. We know what symptoms kind of appear and how patients might look different to a doctor than healthy people do. But that doesn't mean that we know exactly what's causing the disease. And what's causing these diseases really comes down to the cells in your body. And we need to be able to look at the actual cells that are affected by the disease. So for example, in Alzheimer's and Parkinson's disease, like I said, we can't really go around cutting people's brains open. That's not really a great way to do research, and not really that safe for the patients to do that. But we do need a way to be able to look at these cells and not just try to approximate these in these model organisms, because mouse brains and human brains are different enough that it matters for those diseases. So there is a way to do this, and this really is where the power of stem cells comes in. All of the different cell types that make up our body, so we have trillions of cells that make up our body, and they are specialized into these different types of cells, like the brain cells that help you to think, your bone cells, your skin cells, your muscle cells, but they all originally start out as stem cells. So there are about 200 different types of these cells that are specialized to perform different functions that we need to survive. They all have the same DNA, but they use it in a slightly different way to carry out these functions. And so we start off, right after fertilization during embryonic development, our cells all start off as these stem cells that have the power to become all of these different specialized cell types. And so these are called stem cells. And so that really kind of makes stem cells the superheroes of the body, because they can become any type of cell in the body that we have. That includes all these different types of cells that I show here and on the previous slide. And they can make as many copies of themselves as you want to make a sort of unlimited supply of these cells. And so this is really exciting, because once you're able to isolate stem cells, you can expand them, convert them into different cell types. And the reason that we're really excited about all of this is because it's recently became possible for us to actually make stem cells in the lab. And this was due to a discovery by a famous scientist named Shinya Yamanaka, who won the Nobel Prize in Medicine. It's the most prestigious prize you can possibly get for this discovery, just 6 years after he made it. That's actually a very short time for somebody. Between 2006, when he discovered it, and just 6 years later, he got the Nobel Prize. Often it's decades later before you see the kind of impact a discovery has on the field. But in this case, it was the impact was so apparent that the committee didn't feel like they needed to wait that long. So what does this mean? This means that from any patient or from any individual, me, you, anybody, what we can do is take skin or blood cells using just the standard process that you use at the doctor's office when they take a blood sample from you. And what we can do is sort of turn the clock back on these blood cells or skin cells to make them into stem cells. And this is using a sort of combination of genes that Dr. Yamanaka discovered, and they're called the Yamanaka factors. There's just 4 genes. And by sort of turning on these genes in the blood cells or skin cells where they are no longer expressed as they become specialized, that means that what we end up getting is these induced pluripotent stem cells. So we've induced them to become pluripotent, and pluripotent means that they have the potential to become many different types of cell types. And so what we can do then here in the lab, and what our scientists here are doing every single day, is to take these induced pluripotent stem cells, expand them, make many more copies of them so that you have a lot to work with, and then converting them into these different cell types that are affected by disease, like the brain cells, like heart cells, and the different types of cells that I was showing you before. And so what this gives us is a really exciting new window into disease. It allows us to get at these cells that are affected in disease without actually having to cut people open and get at all their organs and take away the cells that they actually need to function. We don't want to do that. But through the power of stem cells, we can make as many of these cells as they want. And they have that person's DNA in them, so they behave the same way that your cells or my cells would behave in our body. So what does this actually look like? We start out with skin cells. This is what skin cells look like. They're called fibroblasts, and this is what they look like under a microscope. They're very pretty. And what we've done here is sort of color them. And what you'll see with all of the cell images that I show is that we're able to color them in with these fluorescent dyes that label different parts of the cell that help us to understand how sort of what the components are of these different cells, how they might differ from each other, and so on. And we can convert these fibroblasts into these induced pluripotent stem cells. And that happens with just the Yamanaka factors. And as you can see, these cells already look very different from the fibroblast, even sort of physically speaking. And then we can convert these stem cells into all types of cells to give us a window into all of these different diseases that I was talking about. And so because of this, this allows us to mimic all of these different kinds of diseases in a petri dish and have a look at what's going on. And so because of this, stem cells are really revolutionizing the way that we do biomedical research. It's a whole new way to understand how cells are behaving differently in disease than they might in a healthy person. So this happens through 3 major ways. One is disease research, like I just said, looking at these cells that are affected in disease, seeing what makes them unique without having to cut them out of somebody's body. What we can also do is take these cells that are affected in disease and test drugs on them just right there in the dish and see how they react. And this is a whole new way for us to discover new therapies that might be effective, and maybe that might be more safe than others. And I'll give you an example of that in a second. And finally, the third way that we're really excited about using stem cells is to make cell replacement therapies. So what happens in certain diseases like Alzheimer's or Parkinson's is that certain types of neurons in your brain are actually dying. And while we don't know exactly why that's happening, and we're hopeful that our disease research efforts are helping us to clarify some of that. We do need to put those cells back at some point. We do need to make sure that the neurons in a patient's brain are helping if we do want to cure them. So one approach that people are starting to use is to grow the cells that are missing or damaged in a disease, and then put those back into a patient to hopefully restore function. And I'll talk a little bit more about each of these and give you some examples in a moment. And so what we do research here at NICEF, I've alluded to a lot of these already, several neurological disorders, including psychiatric disorders, which I didn't mention earlier. We also recently started a program on women's reproductive cancers. So things like ovarian cancer, cervical cancer, where treatments have been a lot slower. We also study diabetes and autoimmune diseases. I had mentioned diabetes earlier. So autoimmune diseases are when your immune system mistakenly sort of attacks different cells in your body or tissues in your body, causing a disease, and that's diabetes is one of those examples. We work on bone engineering to grow bone in the lab out of stem cells. We work on retinal disease. I'll talk about that in a moment. And we also look at rare disease. And so rare diseases are those diseases that affect a very small number of people individually. So these diseases are really difficult because they often don't have models and we don't know very much about them at all. But with the power of stem cells, we can make those models relatively quickly and start to gain insights into what's going on in these diseases for these patients. And that's really great because these diseases really often affect children, which is really tragic. And so the types of brain cells that we can look at that I wanted to talk about first are sort of four major classes of brain cells that make up your brain and allow them to function. I'm sure you've all heard of neurons, and these are the cells in your brain that sort of communicate with each other. They have these synapses and they talk to each other. They send electrical pulses to do that. You have oligodendrocytes, which are sort of the insulators of the brain. So they actually wrap a sheath sort of insulation around these synapses, kind of like your cell phone cables and stuff like that, to allow them to safely transmit those signals. There's also the microglia, which are the kind of patrollers. They're the immune cells of the brain. They kind of run around looking for things to see if there's signs of danger that they need to pick up on, and then you also have the astrocytes, which work together with the microglia, and they collect the waste. If there's any sort of debris or anything that's being generated, then the astrocytes kind of gobble it up. So what astrocytes actually look like, these are some really pretty astrocytes made here in the lab by our scientists out of stem cells, and they, as you can see, they take on these very interesting shapes to sort of branch out and be able to detect any waste and gobble it up as they find it. These right here are what oligodendrocytes look like, what you see in yellow. You can also see those branching out, and they kind of grab onto neurons and create this insulating sheath that I mentioned. The microglia are the ones that are sort of running around the brain. So you can see here that they really do kind of run around. They change shape. They try to sense what's going on in their environment and kind of just make sure everything's okay in the brain. So that's a really important role as well. And then finally, this is what neurons look like when we make them in the lab. They do actually grow out these synapses and reach out to each other and communicate with each other. And they fire these electrical impulses as a form of this communication. And just as an example of sort of how we can start to understand what goes wrong in disease, we've looked at neurons that are made from Alzheimer's patient cells. And what you can see here is that these are different neurons, and they're connected by these synapses here. And what you can see is the way that they're flashing. That's electrical impulses that are being sent to each other. And if we look at these patterns in cells made from an Alzheimer's patient, we see that they're actually not functioning as well. They're not, as frequent. They're not quite as pronounced as you'll see in healthy neurons. And so this is really important to help us understand, in which ways the neurons aren't functioning properly, because that can help us to start to understand how to correct what's going wrong in diseases like Alzheimer's. And the final way that we look at the brain is through what's called organoids. And these are sort of mini organs, and where we put together all of these different cells. So what I've shown you so far are just sort of very 2D, cells growing on a flat surface. And they do sort of reach out and talk to each other. But if you really want to understand how the brain works in a dish, you need to look at how these cells are all talking to each other in a three-dimensional context, and how they're interacting, how they're communicating, how the microglia are sort of running through and around. And so this organoid technology, which was really only developed in the last few years, is a really powerful way for allowing us to do that, and to understand how the brain is functioning in kind of a 3D level. It's still very, very small. You can, they're maybe like this big. You can kind of see them with the naked eye, but it's a closer approximation to what our brain is actually doing. And so this is very new, like I said, and we're starting to learn a lot of new things from looking at models like organoids. The next thing I want to talk about is clinical trials in a dish. So I talked about how drugs have to go through clinical trials so that we can be sure that they're safe and they're effective to use in patients. And so what we're excited about using stem cells for is to find drugs that are more likely to be safe and effective when we use them in actual clinical trials in actual patients. And so what we're doing specifically is to test drugs on the specific types of cells that are affected in disease, just simply by adding them to the dish where we're growing these cells. So one example I wanted to show you all here is cardiomyocytes, and these are the muscle cells of the heart. They contract, and as you can see here in this video, they do actually beat in the dish the same way that your heart beats in your chest. They're able to do those to perform that function. And what happens with heart cells is that sometimes they are affected adversely by different drugs. And you might have heard of certain drugs that cause what's known as an arrhythmia in patients. And this is an irregularity in heartbeat, and that can be caused only for certain people, and we don't really understand why. We're still trying to figure that out. But what's really exciting about being able to make these heart cells in a dish is that we can test whether certain drugs just in a dish will be cardiotoxic. That means causing these issues in heart cells for a given person. If we look at how that person's heart cells respond to the drug in a dish, we do actually see that the arrhythmia that they experienced when they took the drug can be sort of mimicked in the dish. And so we're hopeful that at some point we're going to be able to get to the point where you can test all of these drugs on heart cells in a dish, and you'll know, okay, this person's going to respond well, this person's not. We shouldn't put them in this clinical trial. You shouldn't give them this drug, so that we can prevent these kinds of adverse effects, and hopefully get drugs through clinical trials that are in a much more effective way and kind of beat out those failure rates. What I also want to talk about is what's on everybody's mind nowadays, which is the coronavirus, or COVID-19. And so we've also started to look at how we can use stem cells here to identify new treatments for this new disease. And so others that we work with in the community have come up with ways to convert stem cells into the types of lung cells that are actually infected by the coronavirus. And so what we're starting to do here in our lab is to make these lung cells here in the lab out of the stem cells that we already have, as a resource to help scientists to study the virus, to understand what effects is it actually having on lung cells, or on even different types of cells, and are there drugs that we can use that can stop the infection, that can prevent the infection, that can maybe stop some of the bad effects that the virus has on these different cells. So this is all very early days, but this is just an example of the types of things that we are trying to do with diseases new and old in stem cells, and we're working with a lot of different people in the community to be able to do this. The final thing that I wanted to talk about is cell replacement therapy. So as I was saying, a lot of different diseases are caused by the death or the malfunction of different types of cells in our bodies. And so we're hopeful to use stem cells to make the cells that are affected and actually replace them in the patient to cure the disease. And so one area that we're working on this with is an eye disease known as macular degeneration. And I wanted to show you what these eye cells look like that we're able to grow here in the lab. They're actually really beautiful. These are my favorite cells because they're so pretty and they have this really, really regular sort of a pattern. And that's what they look like when we grow them in the lab. And these are cells that don't directly confer your vision, but they're very important for supporting the cone and rod cells that do actually allow you to see. And so we're working here towards a treatment for macular degeneration. And macular degeneration is a disease that is, it causes sort of loss of central vision. So this is what it looks like. If you see a normal image here or a normal, sort of vision here, this is what happens when you have a loss of central vision. Just what you're looking at right in front of you sort of becomes blurred out. And it's unfortunately, growing in prevalence. It usually affects older people. And is a leading cause of blindness worldwide. And so we're developing a therapy right here in the lab to make the cells that die retinal pigmented epithelial, that just the cells that I showed you on the previous slide. And we're actually building a special facility right here within our institute to make the cells in a way that's safe to put into people. So the other cells that I've shown you, those are not cells that are grown, you have to grow themselves very carefully, obviously, with a lot more regulations and protocols put into place to make sure that they're safe to put into people. And so we're starting that with macular degeneration, but we're really hopeful to expand that beyond into some of the other diseases that I talked about. So one other aspect of disease that I want to address that I'm sure a lot of you are familiar with is the idea that everybody's experience of disease is different. And drugs are also affecting people differently. So we've seen this, and I talked about this with cancer, for example. You can't really do a one-size-fits-all approach to medicine. This doesn't seem to be working, and we're recognizing this more and more in the research and medical side. And so if we really want to understand how people differ in their responses to diseases and drugs, stem cells just really give us a great opportunity to do that. And if we want to really do that properly, we need stem cells from a whole lot of different people to really look at them in the dish and understand how each patient's experience differs and how we need to treat it in the lab. But the problem with that is, that's a lot of work. So this is one of our colleagues here working with stem cells, and so he has to kind of manually do this, moving these cells and these, food for the cells around. And it takes a really, really long time. And we realized very quickly that this was going to take forever if we really wanted to do it at the scale that we're looking for. And so what we've done here is to put together a whole lot of robots to help us do this faster and easier. And this is a video of what some of the robots do here. And so, for example, what you saw in the first video was our scientists working with this sort of 6-well plate. So each well here can be used to put in cells from a different person. So you can do 6 at a time in what he was showing. And what we're able to do with this robot, and what you'll see on the screen right now, is that we can do things like 96 wells. This is 8 by 12. We can do 96 at a time, or even 384 at a time. This allows us to go way faster and way larger. And by using robots, we're also able to reduce the amount of human error that goes into these things. So this has really helped us to do a lot more and to help out a lot of other labs that need stem cell research in their workflows. So that's been a really exciting thing, and I hope to be able to show you those robots in person sometime when we're able to have you here again. The final thing I want to touch on is the idea of artificial intelligence. And I know that a lot of you have probably heard of this, and we're starting to figure out how to use this here for drug discovery. And so what I've shown you are a lot of these beautiful microscopy images of different cells. And so we've collected an enormous amount of data from these cells and these images of both healthy and patient cells. And sometimes it's difficult to see what actually makes them distinct, just looking at it if you're just eyeballing it. But artificial intelligence really comes into play here because it can detect patterns that we can't detect using, the naked eye, or using even computer algorithms that we traditionally use in biology. And so what we're using artificial intelligence to do is sort of figure out, okay, what are those key features that make healthy cells look different from patient cells? And are there drugs out there that might be able to help change those differences that, again, we can't even really perceive? It's a really exciting avenue. It's early days for that, but we're really excited to see what the potential is there. So with that, I'll close. I think the one thing I'd like you all to take home from this is that we really believe that stem cells are the future of medicine. There's so much potential for what they can do to advance research and help us, figure out how to treat your disease with your cells, whether we use those cells or other drugs that they help us discover. There's sort of limitless potential. So with that, I want to thank everybody who's helped to make this possible. Like I said, we have a lot of great scientists and engineers who work together every day. I'd also like to thank the Stavros Niarchos Foundation, the Lori and Tish Illumination Fund for supporting our education and outreach work. And I'm looking forward to all of your questions.
Thank you, Rekha, for teaching us all about stem cells. So we do have some questions, and anyone that has a question, whether it pertains to the beginning of Rekha's presentation or the end, feel free to submit them in the Q&A chat. So we have a question from David. If it is so inaccurate to use animal models, how come some researchers still do?
That's a great question. And I want to be clear that animal models have been, incredibly valuable. Most of what we know about biology and how cells work and how DNA works and how our bodies work really does come from studying these model organisms in a way that's not possible to do, with humans. And so they have been really valuable for teaching us a lot about this. But the similarities between us and animal models do end at a certain point. And for certain diseases, they've been more valuable than others. And it really just comes down to where do those differences start between us and the animal models. And when it comes to medicines, especially, I think that's where things like stem cells have a lot more potential. But that's a great question. And, they're both, I would say they're both valuable, but stem cells are, taking it to another level.
Great. You talked a little bit about organoids in your presentation, and I know they're probably a new word to people. So could these help with organ transplants? Are they full organs in the dish?
Great question. The name organoid does definitely come from organ and the idea that it's, sort of a mini version of that. But I do want to emphasize it's a very mini version of that. So like I said, the brains that we're growing, we're not growing brains, sorry. I want to correct that. The brain cells that we're growing into organoids in the lab are, about this tiny, which is quite a bit smaller than your actual brain. And it's a very, sort of simplified approximation of what happens in the brain. We are not yet able to grow full organs in the dish. It would, there would definitely be some value for that for things like diabetes, where, if you can grow a pancreas in the lab for people whose pancreases aren't working well, or, sort of bypass this whole organ transplant difficulties. There's always issues with your body rejecting transplants, but we're not quite there yet. Right now, it's mostly valuable to, as a research tool to be able to understand how these cells work, and in some cases, it's starting to be valuable for cell replacement therapies, like I described.
We have a question that maybe you can piggyback off of. I'm not sure. I'll let you answer that. But from Pooja, how can we use stem cell replacement therapy in Alzheimer's disease?
Great question, Pooja. So like I had mentioned, in Alzheimer's disease, we've seen, and others have seen, certain defects that are happening in the neurons in terms of the way that they signal to each other. But it's a bit more of a complex story in Alzheimer's disease. There are also the other types of brain cells that I mentioned, like astrocytes and microglia. They appear to be playing an important role as well, and causing some of the dysfunction that we see. So it's a little bit more complicated. I mean, cell replacement therapy is most straightforward when you have a very specific type of cell and you know that it's dying and you just need to sort of put it back. So that, in cases like macular degeneration, we know that cell type. In the case of Parkinson's disease, for example, it's the dopamine producing neurons that are in the midbrain that happen to be dying. With Alzheimer's, we don't have quite as much specificity on that. However, that doesn't mean that it's out of the realm of possibility. There are certain avenues of cell replacement therapies being pursued for that. And I think if you do want to learn more about that, we actually hosted an event about this in January, where the head of our Alzheimer's research program talked about some new routes to cell replacement therapies in Alzheimer's disease. We can post that link and send that around for you, and the videos online.
Great. And someone followed up on that question while you were answering it from Robert. And how about cancer? Can you use stem cells to treat cancer?
Excellent question. Probably the most common stem cell therapy that you all have heard of that maybe you don't know is a stem cell therapy is the treatment for leukemia, bone marrow transplants. And that's often used to treat leukemia because leukemia is a cancer of the blood cells. And as certain blood cells are dying off, if you replace the bone marrow, which contains stem cells for your blood, then your blood has a new source of healthy stem cells. So that's one way in which stem cells can be used for that. Another way that we're using stem cells to study cancer, I'd mentioned that we were working on women's reproductive cancers. And so what we can do is use the sort of principles that we've learned from stem cell research to create sort of tumor organoids. And now these are organoids made directly from a tumor sample that allow the tumor sample to sort of live forever and keep making copies of itself in the lab. And so that allows us to do research on it, to do drug testing, because once you get a tumor sample, you've got a pretty limited supply. So tumor organoid technology is really becoming really important for that. And we do have an upcoming event focused on tumor organoid technology that'll be on June 4th, and hope you're able to attend that if you'd like to learn more.
Yes, if you don't see it on our website, it should be up in the next day or so. So we'll make sure that you please register if you're interested. How long can you keep stem cells in a storage facility?
Excellent question. Pretty much forever. We have ways of storing stem cells. We store them in liquid nitrogen to keep them very, very cold. And they can be preserved essentially infinitely. So that really is a valuable tool that we have to be able to store, to kind of biobank them. And we have a huge biobank of stem cells from hundreds of different people and conditions here at NICEF.
Cool. I've seen them. It's very, very cool.
It's very cool. I hope we can show you sometime too. The liquid nitrogen also looks really cool.
So we have a question from Kathleen. Is cord blood still being used?
That's a good question. Yeah, so we're not really using cord blood here at NICEF. That was it was being used at some point as a source of stem cells, both for therapeutic purposes and for research. There are definitely labs who still work on that and collect cord blood, but that's not an active area of research here. Yeah, because in induced pluripotent stem cells, you can make those from anybody. So that's the kind of the advantage, is that we don't have to wait for cord blood or wait for that person to get pregnant. So definite benefit there.
If a scientist cures a disease in a dish, or they believe they have, does that mean that they cured it for humans, or they cured it, like if I had cancer, would that mean they cured it if they cured it in a dish?
I hope that will be true one day, but unfortunately that's not how it works just yet. What we want to be able to use stem cells for, and what we are using stem cells for, is to predict what drugs are going to be most likely to be effective in a person if we try them in clinical trials. So like I said, because clinical trials are failing so much, we hope that this will be an additional piece of information that we can give to people designing clinical trials, so that they can design them in a way that they're set up for success, and they don't have to spend as much money on them, and the drug gets through much faster. We know right away kind of what's a good candidate.
How long does it take to make a stem cell in other cells like neurons and heart cells?
Great question. So for the stem cells, so doing that whole Yamanaka factor reprogramming method that I showed earlier on, That takes on the order of a couple of months to go through all of that process in the lab. And also we do a lot here to do quality control of the cells to make sure, nothing's gotten mixed up, that they are really pluripotent. So they are able to become all of the different types of cells that we want them to become. We look at the way that they're expressing genes, and if they're doing it in the way that is expected that a stem cell would do, that they do actually, that nobody mixed up the samples and they are actually coming from the person that we thought they came from, that there's no other damage that's accumulated to the DNA or to the chromosomes as the cells have been replicating. So once we go through all of that, it's closer to 6 months or so to make the stem cells out of blood cells or skin cells. And when it comes to making stem cells into all these other different types of cells, there's a really wide range. It really depends on the cell type. So for things like lung cells, for example, the ones that we're working on making now in COVID, they take something like 29 to 30 days in the lab. Other cells like the brain organoids, those can be up to a year or hundreds of days that you have to grow them before they really get that big, and before all of those different types of cells coming up. And we and many others around the world are working every day to make all of those faster, because obviously the faster you can do that, the faster all of this research will go.
Right. Have any medicines been discovered using stem cells that have gone on to be used in humans?
Great question. We hope the answer to that will be many, many of those have been discovered. There is definitely a couple of them that have been discovered using these kind of, trials in a dish sort of thing, and are in clinical trials now. But none that I'm aware of that are actually being used in people at this time. Of course, what you can do is to test drugs that are already approved on the cells in a dish, and see which patients might be better responders than others, or which patients might have adverse effects, like I had mentioned with the cardiomyocytes, and that can help to sort of inform who might be good or bad candidates for certain drugs.
Well, lots of hope.
But that's not yet at the stage of being used in clinical decision making, I should say. It's a very young field. So stem cell, being able to do this, like I said, it was first discovered in 2006. That's only 14 years ago. So everything is still very early, but that is also what makes it exciting, because there's still a lot of potential to be realized.
Yeah. Lots to look forward to.
Yeah.
I'm going to do one last attendee question before we close out. So a question from Shreya. Can you use stem cells to help with finding a cure for allergies?
Good question, Shreya. So we hope so. We don't know of anybody who's actually done anything like this yet, but as I mentioned, one of the cell types that we're able to make with stem cells are the immune cells. And the immune cells are often, what are sort of overreacting when it comes to allergies. And so, that's an area that we and others are looking at, is how to make these immune cells and how to make sure that they really, well, one of the challenges with all of these cell types, I should say, is, you can make them, but making sure that they're fully functional, that they behave exactly as they would in the body, and that they're, as mature as they would be in an adult body, is definitely a challenge. And with immune cells, of course, they acquire this sort of memory as you go through life and you become exposed to different pathogens, and a lot of it is how they behave is shaped through that. So being able to mimic that is definitely more complex in a dish and in a lab environment than with some of the other cell types, but it's definitely an active area of research.
So I'll ask you the final question that I ask all of our researchers that do our virtual classrooms. So what makes you most excited about or for the future of stem cell research?
Yeah, so like I said, I'm a geneticist by training. I went into science because I was really excited about, trying to figure out how genetics make us who we are, and how, my genetics might make me develop different diseases than you, Ali, or, any of you, but hopefully we develop none of them. But, we do have different risks, definitely, that our genetics shape. And I got into it because I thought, well, genetics are the way that we're going to figure all of this out and really figure out how to cure these diseases. But So I did my PhD in genetics, and the more you learn, sort of the more you learn that you don't know. And genetics are a very, very important piece of the puzzle, but how your genetics play out in your cells is really what it comes down to. What comes down to how you function as a human. And so stem cells are really this invaluable tool, I think, for taking genetic research to the next level and really understanding how genetics manifests in your body. And so I'm just really excited about all the new knowledge that we can gain by combining genetics and stem cells. So.
Well, thank you. And thank you so much for teaching us all about stem cells today. I just put a link in the chat. If you have more questions for Nice of Scientists, please feel free to submit them to the link. We'll have some of our researchers try to answer some of those questions for you and email you back. I want to be conscious of everyone's time. I know it's past 4:15, so we hope everyone had a great time this afternoon. Be sure to check back to nicef.org for more virtual classrooms featuring some of our awesome scientists. There will be a brief survey right after the webinar ends. We would love to hear your thoughts and feedback on new topics that you want to hear from us about. Don't forget to tell your family and friends all about what you learned today, and we hope to see you soon. Bye, everybody.
Bye, everyone. Thank you.